Good afternoon. Thank you for joining us for Astardi Medical's webinar presented by Dr. Marc Masor, The Challenges of Preterm Infant Formula Development. I'm Tammy Jansen, co-founder and CFO of Astardi Medical. For those of you who don't know us, Astardi Medical is a precision nutrition company providing digital solutions to support nutrition and feeding practices for preterm infants in the NICU. We're hosting a series of educational webinars this year, all focused on the growth and development of preterm infants and the importance of feeding, nutrition, and gut health. I'm thrilled to share our incredible lineup of upcoming speakers with you. You can find the details for all of our webinars, both past and future, on our website. Please mark your calendars and join us for our next webinar on November 17th for a presentation by Dr. Richard Chandler on human milk benefits for the preterm infant. Dr. Chandler is Program Director for the Fellowship in Neonatal Perinatal Medicine at Cohen Children's Medical Center and Northwell Health in Long Island, New York. Nutrition in the NICU is his major research and clinical interest. He's conducted studies focusing on the effects human milk has on the recipient premature infant and how processing affects the composition of human milk, and he has over 100 publications in this area, so please mark your calendars. So we'll get started. We'll have plenty of time for questions at the end. Please feel free to post your questions throughout the presentation in the chat box on the right side of your screen. We'll compile the questions and get to as many as we can. I'm pleased to introduce our esteemed presenter and expert in the field of neonatal feeding and nutrition. Marc Masor, with a Ph.D. in nutritional biochemistry, retired as Director of Clinical Nutrition Research at Abbott Nutrition after spending 16 years conducting infant nutrition research. He led the research project for the development of Similac Advance and is an inventor on 10 patents covering Similac infant formulas. He's spoken to physicians, nurses, dietitians, regulatory agencies, and conferences across the US and in over 30 countries. You might be interested to know that Dr. Masor's PhD research project was to determine the effect of dietary protein on lactation. Dr. Masor has available a complete list of references for this talk for those of you who'd like one. I'll now turn it over to you, Dr. Masor.
Okay, just checking that you can see my slides.
Yes. Not yet.
Okay. How about now?
Not yet.
Okay. I'm not sure why it's not showing. How about now?
Okay, now we can see them. Yep.
Okay. All right. Well, welcome everyone. I'm talking to you from my home in Durango, Colorado. Nice little mountain town down in the southwest corner of the state. This is a view up the Animas Valley. This is the campus of Fort Lewis College, the campus in the sky. A lot of people come here to ride the Durango-Silverton Railroad, narrow gauge railroad that goes up into the mountains. Nearby is Mesa Verde. These are the cliff dwellings of the Anasazi, who were the ancestors of the Pueblo Indians. Summer here, it's quite beautiful, brilliant blue skies, bright sunshine. Now, that's my front yard. This is what it looks like in the winter. We do get a little bit of snow here. That was perhaps a bit too much that year. I'm going to talk to you about the challenges of preterm infant formula development. And before I go on, I will disclose that I am currently a paid speaker for Abbott Nutrition, but I'm not a stockholder for any manufacturer of infant formula. And what I'd like to cover to different extents are infant formula regulations, considerations in the manufacture of infant formula, and also looking at nutrient composition, which is really a discussion about how a company selects ingredients to put in formula. So infant formula is regulated by the Infant Formula Act, first passed in 1980, updated in 89, has not been changed since then. And it specifies minimum nutrient requirements It defines adulteration, establishes nutrient and quality control procedures, prescribes recall procedures, and specifies inspection requirements. The Infant Formula Act specifies a minimum of 20 nutrients and a minimum and maximum of another 9 nutrients. So naturally, all infant formulas are going to have similar nutrient levels because they have to comply with the Act. The nutrient label that you see on formulas specifies nutrient levels, and they must be at or above the label claim by the end of shelf life. And all ingredients that are added must be listed, and they're placed in order of predominance, similar to what you would see on other products. There are some infant formulas that are exempt from all the regulations, and those would be formulas for infants who have an inborn error of metabolism, like ketonuria, low birth weight or prematurity, and then unusual medical or dietary problems such as food protein-induced enterocolitis. So premature infant feedings must be liquid, and that's because the process of producing powder, whether it's spray drying or dry blending, doesn't get to the same internal temperature as a liquid. And because of that, Coronabacter Sasaki spores will survive that process. And then about 15, 20 years ago, there were a number of cases of C. Sakazaki sepsis and death. And because of that, the FDA proscribed the use of powder in the NICU unless no other alternative was available. So manufacturing is a fairly complex process, partially because liquid formulas are actually sterile emulsions, and that sterilization process may affect the stability of that emulsion. In addition, sterilization may affect nutrient stability or the bioactivity of nutrients or other ingredients in the formula. Shelf life validations are actually done in real time. In other words, a 12-month label claim requires storing the formula for 12 months and then measuring the nutrient levels at the end of that 12-month period. And it's just not just nutrient levels, but also formula stability and sterility also have to be guaranteed at the end of shelf life. Let's think about the ingredient selection. So over the years, research really evolved from simply copying the composition of breast milk to looking at the developmental physiology of breastfed infants. Instead of just looking at simple parameters like growth, following developmental and clinical outcomes. Because we recognize that you can't really copy breast milk. Formula is made of ingredients, not nutrients. You may want to add an essential fatty acid. You have to find a vegetable oil that contains that fatty acid. And of course, manufacturing and processing can affect the bioavailability of nutrients. Breast milk is a living tissue. It has living cells, active enzymes, hormones, growth factors, immunoglobulins. Well, these aren't ingredients that you can pull off the shelf and put in the formula, and most would be destroyed by processing. But what you can do is you can test ingredients in clinical studies to see if you can match the developmental physiology of breastfed infants. Now, you might think that there are a lot of experts out there giving guidance, and there are. For instance, the Life Science Research Organization is a panel of experts hired by the FDA to look at these kinds of questions. The American Academy of Pediatrics has a committee on nutrition, as does the ESPGHAN, the European Society for Pediatric Gastroenterology, Hepatology, and Nutrition. And then there's the FAO and WHO with the UN. But for instance, here are the recommendations for some of these groups for the addition of DHA and arachidonic acid that were being made at the time that companies were actually developing these formulas. And you can see that their recommendations were pretty broad, and quite a few of them even at that time, said there was really insufficient evidence to make a recommendation. So it's really up to the formula companies to do the clinical research to determine what levels are appropriate in their formulations. In order to bring an infant formula to the market, really what the FDA requires is a growth study, and that's simply looking at weight, length, and head circumference. But there are other aspects that are important to consider. Tolerance studies are really a key part of this development, and that's looking at things like stool characteristics, the incidence of spitting up and vomiting and crying. And this is generally the first study that's done, because even if you had a formula that prevented otitis media, if infants couldn't tolerate it, you wouldn't be able to put it on the market. For example, for the research that led to the development of Similac Advance, it was a major project with over 50 clinical studies, over 6,000 infants. About 1,100 of them actually were fed the final formula. And of those 1,100, 100 of those were intolerance studies, because that really is a key part of the development. Nutritional status can be assessed with balance studies, looking at blood metabolites. Efficacy studies are generally done when a company wants to make a claim, and they have to have an efficacy study to support that claim. In premature infant studies, growth is really of primary importance. When do they regain birth weight? Do they improve across growth percentiles, which is what one would like to see, and make sure they're not going down in growth percentiles? Every adverse event that occurs during a premature infant study is documented, recorded. Things like neck and late onset sepsis. Feeding is followed, time to full feeds, what's the intake, and then various efficacy outcomes such as length of stay. But there are a lot of challenges in premature infant studies that go well beyond what you see in term infants. First of all is subject availability. There simply aren't that many preterm infants available for these studies. And it's a very heterogeneous population. Term infants aren't exactly like laboratory rats, but they're much more homogeneous than premature infants. At any gestational age, you could have small for gestational age, large for gestational age, all kinds of comorbidities that may influence subject selection, and how many NICUs are available. And the DHA study that we did, it took 15 NICUs in 3 countries to get enough subjects to do the study. Conformity to the study protocol is always a challenge, because consider a neonatologist who has an infant in a study and the infant is not doing too well. It would be almost unethical for them to conform to a protocol when they recognize that leaving that protocol would be better for the infant. Then there's also the issue of consistency of measurements of the variables. Different NICUs are going to have different abilities to follow those variables. They may use different labs that could have variations in the measurements. And all of this, of course, leads to what may be very complex statistical analyses to account for all of that variability. Then there are what I call hiccups in the process. So let's say you defined a formula and you send a clinical product request and they make that formula. Then you initiate a tolerance study to make sure it's well tolerated. And meanwhile, product development is working on optimizing the formulation. But suppose in that tolerance study, there are tolerance issues. Then one has to ask, well, is that because of an ingredient or is it a processing parameter? And regardless, that sends you back a step. So you kind of have to start the process all over. Now, if the research model for term babies is the developmental physiology of breastfed infants, what's the research model for preterm infants? Well, there really isn't one. They're supposed to be in the womb. What we do know is that regardless of gestational age at birth, these infants lose weight, and it may take 2 weeks before they regain their birth weight. And in the meantime, they may have fallen from the 50th to the 10th percentile. In other words, they're behind in growth. This is looking at weight gain in the NICU based on gestational age. So 24-weeker may be gaining about 5 grams a day, 30-weeker maybe 15 grams a day. If they had remained in the womb, this is what their weight gain would have looked like. Regardless of their gestational age at birth, they're well behind what they would have been if they had remained in the womb. This is a study from the Vermont-Oxford group asking the question whether poor weight gain in the hospital increases the odds for poor outcome 20 months later. A fairly large study, 490 infants, less than 1,000 grams. And to answer that question, they compared the infants in the lowest quartile of growth, 12 grams per kilo per day, to those in the highest quartile. And compared to those in the highest quartile, those in the lowest quartile had a 2.5-fold greater risk of neurodevelopmental impairment, a 2.25-fold greater risk of low Bailey scores, less than 70. That's 2 standard deviations below the mean, and an 8-fold greater risk of cerebral palsy. Ernst and colleagues looked at the relationship between protein intake and weight gain in 69 infants, less than 1,000 grams, and they showed that 45% of the increased weight gain was associated with protein intake. Aaron Krantz also looked at the effect of poor head circumference growth in the hospital and how that affected the odds for poor outcome at 20 months, the same infant population. They also compared the lowest quartile to the highest quartile in head circumference increase. And as what they saw with weight gain, there were significantly increased odds for poor outcome for cerebral palsy, low Bailey's, and neurodevelopmental impairment. And similarly, looking at the relationship between protein intake and head circumference growth, Ernst showed that over half of the increased head circumference gain was associated with enteral protein intake. Embleton looked at how protein intake led to a cumulative deficit of protein. And this was based on the protein recommendations at the time the study was done. And we see that for the larger preterm infants over 31 weeks of age, even though protein intake was increased, the deficit did not improve. And with the smaller babies, that deficit got even worse. And he showed the same thing with weekly energy intake and the cumulative deficit. Again, the larger infants, no change in the deficit, and with the smaller infants, the deficit continued to get worse through 7 weeks. And of course, that led to a significant loss in Z-scores. A Z-score of 1 is about 1 standard deviation on the growth charts. And here with the larger preterm infants, they're losing about a standard deviation in Z-score, and for the smaller ones, even more. So this is a significant problem dealing with achieving appropriate growth for preterm infants. And this is a study looking at how gestational age affects brain development. These MRIs were all taken at 40 weeks gestational age. The picture on the left is an infant born at 23 weeks gestational age. This is a 30-weeker, and here we see a term infant. Look at all the space in this 23-weeker and the small size of the brain, the lack of size and convolutions in the 30-weeker compared to a term infant. The brain is highly susceptible to deficits in growth. There's a huge amount of growth in the brain. In just the last 5 weeks of gestation, myelination increases 5-fold. And this is a study that just came out this year looking at how gestational age at birth affects brain morphology at 10 years of age, a surprising outcome. Looking at the total brain volume at 10 years of age compared to gestational age at birth, you see this direct relationship. This is based on over 3,000 subjects. And this is corrected for all of the variables that are known to affect brain development, including gender, age at the time of the assessment, maternal ethnicity, age at intake, marital status, educational level. And what they found was that every week of gestational age, or every week that the infant remains in the womb, accounts for 4.5 cubic centimeters increased size in the brain. And even for late preterm or early term infants, as they're now described, some of them, the same relationship. Every week of gestation leads to a 4.8 cubic centimeter increase in brain volume 10 years later. Pretty remarkable. Nowadays, the standard practice in NICUs is to feed human milk. The challenge is that either with mom's own milk or donor milk, it must be fortified to meet the growth requirements of preterm infants. Human milk is insufficient in protein, calcium, phosphorus, and a number of other nutrients for preterm infants. So a major question is, how does preterm formula compare to an all-human milk diet? And this question was answered by Christopholo when he fed infants either a preterm formula or donor milk plus a human milk-based human milk fortifier. There was no difference in weight gain between these groups, nor in head circumference. There was in length gain. Infants fed the preterm formula had significantly greater length gain, but I always caution looking at length gain. This is the least accurate of all the anthropometric measurements. Even in studies with term infants, when study monitors would train study personnel how to accurately measure length, we still would see occasionally shrinking babies because it can be very challenging, and even more so with preterm infants. What really caught the researchers' attention was how the different feedings affected necrotizing enterocolitis. Neck surgery, or neck and or death was significantly greater in infants fed the preterm formula. So is that because of something in the preterm formula that increased the risk, or is there something in human milk that had a protective effect? Many people thought that the protein, basically being camel protein in preterm formulas, may be responsible for that. And the thought was that if one hydrolyzed that protein, it would reduce the antigenicity of the protein and help prevent NEC. This is a study done by Maggio et al. In Italy, looking at feeding either a standard preterm formula or a hydrolyzed formula. And these are the anthropometrics at birth, no differences in weight, head circumference, or length. And this is a very small study. It was just to get a first look at the potential effects of hydrolyzed formula. But they did see some pretty dramatic changes. Looking at urinary essential amino acids, they saw that by 14 days of age, infants fed the hydrolyzed formula had a much greater loss of urinary essential amino acids. And that led to differences in growth. Here again, looking at Z-scores for weight, the infants on the hydrolyzed formula were losing ground in Z-scores, whereas the intact fed infants maintained their Z-score. And we see in a more dramatic effect in head circumference Z-scores, where they decrease in the hydrolyzed protein and increase with the intact formula. And of course, if they're losing Z-scores in head circumference, that doesn't bode well for cognitive development. Rigo and colleagues looked at using hydrolyzed protein in preterm infants by assessing nutrient balance, and they saw that both for nitrogen absorption, nitrogen retention, and phosphorus absorption, there was a significant decrease with the hydrolyzed protein infant formula. And this led to the question of do these things really, do these hydrolyzed formulas really change the incidence of necrotizing enterocolitis? This is a recent study, a meta-analysis, 4 different studies, and here you see the weight given to each study depending on how well the study was performed. And it's pretty clear that overall there was really no benefit to using hydrolyzed formulas in preventing NEC. And that shouldn't be surprising because when you look at all of the factors that predispose the premature infant to NEC, they're quite extensive and complex. They have decreased digestive ability, less gastric acidity, lower levels of digestive enzymes. The mucosal coat is not as well developed. There are fewer goblet cells. They have an immature gastrointestinal lymphoid tissue. Many preterm infants experience dysbiosis in their microbiome. Peristalsis is slightly impaired. They have lower motility. Altered tight junctions lead to increased permeability, meaning they're more vulnerable to potential toxins, bacterial toxins getting into the bloodstream. And there's an increase in toll-like receptors, which can mean increased levels of inflammatory cytokines at apoptosis, decreased mucosal heating, and lower circulation. Then there are unexpected ingredient interactions. Probably the best documented of these is the effect of palm oil. Companies use palm oil in order to match the palmitic acid content of breast milk, but the triglyceride structure of palm oil or any vegetable oil is much different than in breast milk. In vegetable oils, the palmitic acid is hydrolyzed, and then it can bind with divalent cations like calcium, forming the salts. And here we see that leads to a significant decrease in both fat absorption and calcium absorption, and as a consequence, a lower bone mineral density. Now, these happen to be 2 head-to-head studies between Similac and Enfamil, but there are now about 10 studies in the literature, not just in the US, but also in Italy and the UK, and all of them, without exception, show that palm oil reduces fat and calcium absorption. 4 of these studies looked at bone mineral content, and all 4, without exception, show decreased bone mineral content. And that leads me to this point. When people look at infant formulas, they say, well, they're all the same. They're all FDA approved. They all have similar nutrient levels. And in fact, they do. In fact, they have to. It's the law. It's the Infant Formula Act. But formulas aren't made of nutrients. They're made of ingredients. They may have the same level of fat, but they use different vegetable oils to achieve that. And those differences in composition, in this case, the fat blend, can lead to physiological differences such as fat and calcium absorption, and that can affect a development outcome, in this case, skeletal development. Formulas are different because they're made from different ingredients, and those ingredients can affect development. Then there are novel ingredients. Novel ingredients are those that have not previously been in infant formula, and they deserve special recognition. Some of them that are in infant formula now were considered novel ingredients before they were added, such as DHA and ARA, arachidonic acid, the prebiotics, galacto-oligosaccharides, fructo-oligosaccharides, human milk oligosaccharides, which are most recently addition, but also milk fat globule membrane, bovine lactoferrin, and carotenoids, particularly lutein and lycopene. Novel ingredients must be designated as GRAS, generally recognized as safe. That's an FDA designation, and that is usually obtained by the manufacturer of the ingredient. So for instance, the GRAS approval of DHA and ARA that was granted to Martech, that took well over 5 years to develop. This is a pretty exhaustive process to be certain that these ingredients are going to be safe when added to infant formula. So let's look at a couple of examples here. So carotenoids are added to infant formula because these are powerful antioxidants. And what makes premature babies' brains and eyes particularly vulnerable to oxidative damage? Well, they have deficient antioxidant defenses. They have lower levels of antioxidant enzymes, such as superoxide dismutase, glutathione peroxidase, catalase. They also have lower levels of basic antioxidants, vitamin E, vitamin C, and the carotenoids, as well as lower levels of iron-binding proteins. Free iron in the blood, iron is a powerful oxidizing agent. Just think of how quickly iron rusts. In addition, there's increased oxygen exposure. Just being born, room air is a 20-fold increase in oxygen pressure in the lungs. And of course, if the infant is ventilated, that increases even more. Light is energy. Light also can cause oxidative damage, and iron ions if they're not bound to these proteins. So this combination leads to free radical mediated oxidative stress that can cause cell and tissue injury. Every major disease of prematurity has oxidative damage as part of its etiology. Bronchopulmonary dysplasia, neck, retinopathy of prematurity, interventricular hemorrhage, or paraventricular leukomalacia. In this study, carotenoid intake was controlled in these groups. Here we see the levels in infants fed control formula. That was what the levels you see there is just what happens to be inherent with the other ingredients. Here are the levels in the study formula, and here in human milk. This is from a reference group. And you see that the levels in the study formula are much higher than the levels seen in human milk. But when we look at the blood levels of infants fed these formulas, we see a much different picture. In this case, again, the control formula, here the study formula, and here human milk. So this is another example of why it doesn't necessarily make sense to match the composition of breast milk, but rather look at the physiological response. In this study with premature infants, Rubin et al. Looked at C-reactive protein, which is a generalized measure of inflammatory state. Here are the CRP levels in infants fed the study formula and in human milk, and here are the control formula without the added carotenoids, significantly higher by 40 weeks gestational age. One can also look at eye function using something called the electroretinogram. This is a very unique procedure in which a contact lens is placed on the eye of the infant, and it has two electrodes which just touch the surface of the eye. Then when a light is shined through the lens, those electrodes can record the electrophysiological response of the retina. The A wave of that response describes the number of rods that respond to the light, and it was significantly higher in infants fed the study formula containing the carotenoids, lutein in particular, which is concentrated in the eye. The saturated amplitude describes how quickly it takes the rods to reach a maximum response. And here again, higher in infants in the study formula. Now, this is not a significant difference, but there was a significant correlation between plasma lutein and the saturated amplitude. Novel ingredients are often tested first in term infants. There are a number of reasons for this. First of all, term infants are less vulnerable than preterm infants. It's a safer way to test the efficacy and the safety of an ingredient. And additionally, if you think that an ingredient that you're adding is going to be beneficial to infants, it makes sense to test it first in the product that's going to affect the largest population of infants, which would be term infants. One of those novel ingredients that has received a lot of attention lately are human milk oligosaccharides. It's a huge component of human milk. There are more oligosaccharides in human milk than protein. And they were first recognized because they act as prebiotics. They are selectively fermented by beneficial bacteria. In some of the older literature, when milk was analyzed, there was a component that was referred to as bifidus factor. Anytime you see the word factor, that means people don't really know what it is. And it's likely that that was HMOs, human milk oligosaccharides. Here is a thin layer chromatography of oligosaccharides. This column is the standard. These are the known oligosaccharides. Now compare human milk to goat, cow, sheep, pig. Human milk is truly unique. The human milk oligosaccharides are more numerous and more diverse than any other mammalian species. HMOs are synthesized in the mammary gland from lactose, and then with a combination of these sugars, a variety of HMOs are produced. These are some of the major HMOs, but depending on what study you look at, somewhere between 1 and 200 unique human milk oligosaccharides have been identified and isolated. 2-fucosyl lactose, or 2FL, is the most abundant HMO in most mother's breast milk. It's present in 75 to 80% of a mother's breast milk. Lactoaminotetrose is another HMO that has been extensively studied and now added to some infant formulas. Obviously, 2FL is not collected from human milk. It's biosynthesized using a non-pathogenic E. coli. Glycerol is used as a carbon source for the production of fucose. Then lactose is added, and 2-fucosyl transferase is inserted into the E. coli gene so that it can then not only synthesize 2FL, but actually excretes it into the growth media, so that when that fermentation is completed, one simply has to centrifuge the cells, and then you can isolate the 2FL from the growth media. Structurally and compositionally, it is identical to the 2FL in breast milk. And if you're thinking that that sounds like a Frankensteinian type of process, consider all of the recombinant proteins that are commonly used and have been used in human populations for years. Human insulin growth hormone, parathyroid hormone. These colony stimulating factors are used particularly in treatment of cancer, as well as these interferons. These biologics are often used for diseases such as Crohn's and rheumatoid arthritis. TNF alpha antibody, for instance, that is Humira. Again, the first thing the FDA wants to see when you add a novel ingredient is that it doesn't affect growth. So this is a growth study with a standard formula, 19-calorie formula, looking at average weight at study visits from day 1 all the way through 119 days of age. These are infants fed the control formula. There were 2 study formulas, one with 0.2 grams of 2FL per liter and one with 1 gram per liter. And the total oligosaccharide content was equalized using the lacto-oligosaccharides that you see down here. So the total content was the same. And what about breastfed infants? Well, they weren't different than any of the formulas. And it wasn't just weight, but also there were no differences in length or head circumference. This showed that 2FL did not impair growth in any way. What you're looking at are different cells of the immune system, and in those boxes, those are cytokines. These are the chemical messengers that immune cells use not only to communicate with each other, but to coordinate their attack against pathogens. It makes sense then that if one wanted to assess some aspect of immune development, that you could do that by looking at these cytokine levels. So in the growth study that I just showed you, researchers took blood samples from a subset of those infants and looked at levels of plasma cytokines, particularly inflammatory cytokines. Here we're looking at TNF alpha. You might be thinking that an inflammatory cytokine might not be such a great thing, but it is a critical part of communication in the immune system. You've probably heard ads for Humira where they advise patients that they might be more vulnerable to certain diseases. Here are the TNF alpha levels in infants fed the control formula in orange, the low 2FL formula in green, and the high 2FL formula in red. This is the average for the human milk group, and this is the 95% confidence interval. The infants fed the low 2FL formula were right where the human milk group was, not different, but significantly lower than infants fed the control formula. There were 4 other inflammatory cytokines that were measured, and the pattern is the same in all of them. The infants on the low 2FL group not being different than human milk and being significantly lower than infants fed the control formula. Researchers also wanted to look at how immune cells would respond to a pathogen. So peripheral lymphocytes were stimulated or exposed to respiratory syncytial virus, and then the cytokine response was measured. Here again, we see TNF alpha, and it's the same pattern. Once again, the infants on the low 2FL formula had a response similar to the breastfed infant. And remember, that's our model, a physiological response like the breastfed infant, and significantly lower than infants fed the control formula. And the same pattern was seen in the other cytokines that were measured. So this is the kind of research that allows scientists to say that the lower level of 2FL is probably the most appropriate one to use. And obviously, looking at those cytokine responses, it's clear that 2FL has effects well beyond simply being a prebiotic. This is one of my favorite studies, looking at how 2FL may attenuate the severity of NEC. This is in a mouse model. Here we're looking at the intestines and a micrograph of the villi, and these were, well, in the paper they said they were fed breast milk. Actually, it was mouse milk. I guess researchers didn't realize that mice don't have breasts. In order to develop necrotizing enterocolitis, the mice were fed a formula of half infant formula, half canine formula. They were raised in a low oxygen environment and exposed to a pathogen. And you can see it was a very effective model. Clearly some necrotic gut there, and the villi are completely disrupted. The next slide you'll see are from mice that were given this exact same treatment, except that in their formula, 2FL was included. And it's a quite remarkable reversal. And of course, you see that's a little more inflamed than in the control mice, but the villi are much more intact than in the NEC model. What I liked about this study, they also looked at mechanism, and they measured what they call the Villus perfusion index, which is a way of measuring blood flow to the gut mucosa. And it's apparent that 2FL completely reversed the inhibition of circulation to the gut. These are studies showing how 2FL actually affects brain response, looking at long-term potentiation. In this study, rats had an electrode implanted into the hippocampus, which is the part of the brain that converts experience into memory, learning. And they initiated a high-frequency electric stimulus to that electrode and looked at the response. Here's the baseline for rats given either control chow or chow containing 2FL. And here we see the response, significantly higher in those rats getting the 2FL in their chow. They followed that up with this experiment, which a beautifully designed procedure. They were looking at the response over 2 days. And as they saw before, putting 2FL in the chow led to a pretty dramatic increase in the amplitude of the response. Then another group in this experiment, they did a vagotomy. And when they did the vagotomy, the response disappeared. So this tells us that 2FL either directly or indirectly interacts with the vagus nerve and affects brain response. It doesn't tell us whether the 2FL interacts directly with the vagus nerve or if it's related to the effect of 2FL on the microbiome, which in turn may affect that. But regardless, a pretty dramatic illustration of how what happens in the gut can affect brain response. Vagotomy canceled the effect of 2FL in these rats. Then there's the question of natural versus synthetic ingredients, and vitamin E is a case in point. Vitamin E has 3 different chiral centers. What that means, remember this is a two-dimensional picture, but this methyl group can either be coming towards us or away from us. And those 3 chiral centers mean there are a possible 8 different isomers. They have the same chemical structure, but a different 3-dimensional structure. RRR alpha-tocopherol is natural vitamin E. The 3 isomers that have the methyl group in this position and the R position have some vitamin E activity, but the other 4 isomers have no vitamin E activity. And synthetic vitamin E is an equimolar mixture of all 8 isomers. When you take synthetic vitamin E, only 1/8 of it is actually vitamin E. The other 7 isomers are synthetic. They do not occur in nature. This is a study to determine if there is a benefit in infants getting natural vitamin E, looking at plasma levels of alpha-tocopherol with infants fed a formula with either 20 IUs of natural E, 10 IUs of natural E, or 13.5 IUs of synthetic vitamin E. And here we see that the infants getting the 20 IUs of natural E with significantly greater levels in the blood than those getting the synthetic vitamin E. Also of interest was the effect on gamma-tocopherol. Gamma-tocopherol is also a potent antioxidant and important in physiology. But because these tocopherols compete for entry into the blood, there's a possibility of interference. And in fact, we see that the 10 IUs of natural vitamin E had higher levels of gamma-tocopherol than the infants fed the synthetic vitamin E, and not different than human milk. And more importantly, perhaps, is looking at the ratio of gamma to alpha tocopherol. And here we see that the infants who received the 10 IUs of natural vitamin E were not different than human milk, but significantly higher than infants in either the 20 IU natural E or the synthetic E. So once again, this is the type of study that leads scientists to determine how much of a nutrient and which nutrient to add. We know that in studies of human milk, about 70% is natural vitamin E, but 30% are these synthetic isomers of vitamin E. How do they get there? Well, if a breastfeeding mother is taking a vitamin supplement that has synthetic vitamin E, or if she's eating foods where the manufacturer supplements with synthetic vitamin E, it gets into her breast milk. And as one might expect, it affects alpha-tocopherol in infants' brains. This is from an autopsy study looking at tocopherol in the frontal cortex, the occipital cortex, and the hippocampus. Here we see the total alpha-tocopherol, and this is how much was RRR, alpha-tocopherol, natural vitamin E. So the difference, these are synthetic isomers in babies' brains. Does that matter? Well, let me give you an analogy. I'm sure you're all familiar with trans fatty acids that come into our diet from partially hydrogenated vegetable oil that was declared GRAS by the FDA over 50 years ago. Then we discovered that trans fatty acids actually increased the risk for cardiovascular disease. So the FDA removed GRAS status. Well, we see the same thing with these synthetic isomers. Well, here's some evidence that we may well see that. This is a study looking at the effect of different sources of alpha-tocopherol in the brain. So these are what are called alpha-tocopherol transfer protein null mice. These mice lack the gene to synthesize the protein that transfers alpha-tocopherol from the liver to peripheral tissues. So there were 4 groups that were fed different diets. These are all the ATT null mice. The control group were normal mice that had the appropriate transfer protein. So this is a vitamin E deficient group with no vitamin E added. This group had 600 milligrams per kilogram of diet of natural vitamin E, and then 2 different levels of synthetic vitamin E. This is the level of the natural vitamin E that made it to the brain. And a couple of observations that even without the transfer protein, the rats without that transfer protein were able to accumulate the same level of alpha-tocopherol in the brain as the control mice. Of course, it took quite a bit more, 600 versus 75. Now looking at the 3 isomers that had some vitamin E activity, we see that it accumulates in the brain of these null mice, even though they lack the transfer protein. And even in the control mice with the transfer protein, they too are collecting these synthetic isomers. And here, the isomers that have no vitamin activity. A pretty dramatic effect. And we see that regardless of a rat does or doesn't have that transfer protein, it's absorbing considerable amounts and depositing considerable amounts of these isomers in the brain. And as for effect, the researchers looked in the cerebellum at myelin-related genes. They identified 2 transcription factors and 3 of the genes that are targets of those transcription factors, 2 of them producing myelin components and 1 myelin formation. The arrows show you the direction in which the transcription factors influence each other or the target genes. And here we see that the change compared to natural vitamin E, a significant decrease in the expression of these transcription factors and the target genes. Now, here is evidence that synthetic vitamin E may well have a deleterious effect when fed to infants. So how do alpha-tocopherol and lutein work together in the membranes of the eye and the brain? This is a cartoon of a bipolar membrane. These are the polar heads of the phospholipids. These are the fatty acid nonpolar tails. This is an artist's depiction of the outer segment membrane. The red dots correspond to the polar heads, and the green squiggly lines, the fatty acid nonpolar tails. The red protein in the middle is rhodopsin, which of course is present in rod membranes. This is a typical DHA containing phospholipid. The fatty acid tail is the nonpolar part of the molecule, and the phospholipid head is the polar part. So if this cartoon represents that phospholipid, when it's inserted into the membrane, the polar head is the polar part of the membrane, and the fatty acid tail is in the middle. And of course, it can be inserted on either side of the membrane. Alpha-tocopherol also has a polar head and a non-polar tail, and when it is inserted in the membrane, it too can be on either side of the membrane. Lutein is rather unique. It has two polar heads, and the center is the nonpolar component. When lutein is inserted into the membrane, it crosses the entire membrane. That probably is what accounts for its remarkable stability in these membranes. And what scientists have found is that wherever you have phospholipids rich in DHA, you also see alpha-tocopherol and lutein. Clearly, they have a role in protecting DHA, a highly unsaturated fatty acid, which is very vulnerable to oxidative damage. Now, I don't really expect you to read this slide. I just want you to be impressed with the key role the DHA plays in intra and extracellular metabolic processes. Let's look again at the nutrient label. The nutrient label doesn't tell you what's in infant formula. To know what's in the formula, you have to look at the ingredients. What I've highlighted in yellow are ingredients that are not on the nutrient panel. The sources of DHA and ARA, 2FL, fructooligosaccharides, the carotenoids. Here we see vitamin E. The only way you know it is natural vitamin E is if it has that letter D, D-alpha-tocopherol. If it's D-L-alpha-tocopherol, or if it simply says alpha-tocopherol acetate, it's synthetic. And then, of course, nucleotides added to formula are not on the nutrient label either. But what I find challenging is that the implications of nutrition research are difficult for a lot of people to accept, even people like you. And I think it's because the developmental effects of nutrition are invisible in individuals. You can't see nutritional effects. With medicine, you do. If an infant has an infection, you give them an antibiotic, you see the infection going away. You can't see nutritional effects. This is the study that led to the marketing of a nutrient-rich discharge formula. These are preterm infants fed either a standard formula or the nutrient-rich formula from discharge through 12 months of age. And what you're looking at are significant differences in head circumference, brain growth, all the way through 12 months. Now, if you fed the preterm infant the term formula, that's the curve you would see. Well, you might think that looked pretty good. What you can't see is that if the infant had the right formula, it would have a bigger brain. The only way you know about these nutritional effects is by looking at properly designed clinical studies, blinded, randomized, controlled clinical studies. And what happens in those clinical studies is what happens in your patients. So in that very direct way, their development depends on your recommendations, because formulas are different. They're made from different ingredients, and that affects development. When evaluating clinical studies, you have to ask, is the study design appropriate? Do they have the right population? Are they looking at the right outcomes? Do they have enough numbers of subjects? Are the claims made by the company justified by the data? Importantly, is the marketed formula the same one used in the clinical study? You can't assume that by adding an ingredient studied in a different formula that it would have the same effect, because formulas have different processing parameters, and that can clearly affect outcome. In summary, the design, manufacturing, and marketing of premature infant formulas is very complex, it's highly regulated, and involves sophisticated product development and analytical development, and requires extensive preclinical and clinical research. Well, thank you for your attention. I'd be happy to take any questions.
Great. Thank you, Dr. Masor. So I'm going to ask you to stop sharing and turn on your video, and we'll take a few questions.
Okay.
All right. So the first question is, are feeding studies for preterm infants necessarily segregated according to birth weight, such that there is a distinction between low birth weight, very low birth weight, and extremely low birth weight infants?
The simple answer is yes. And typically you see in a lot of preterm studies, they'll be separated by over and above a certain birth weight, or sometimes into 3 different ranges. And again, that's one of the complexities, is trying to manage the differences in gestational age and how that affects growth. So yes, that is generally taken into account.
Okay. The next one is, how do HMOs vary in mix and amount as mom's milk matures, and are they similar in mix and amount in donor milk?
The HMO composition of breast milk is genetically determined. They're actually related to blood types. So these are not generally affected by diet. They're individually affected by the genetic makeup of the mother. So for instance, women who make 2FL are called secretors, whereas there are women who do not make 2FL, they're non-secretors. So there are differences, but all human milk is rich in HMOs, but the exact composition can be different between different mothers.
Okay. Next question, is the effect of 2FL in the reduction of neck severity due to its effects on the microbiome, or more a direct effect on intestinal immune cells?
Well, we don't really know. That question hasn't really been answered directly. We do know that the microbiome has a profound influence on immunological development. There is direct communication between microorganisms and cells of the gut immune system. The GI tract is the largest immune organ in the body. 70% of the immune cells in the body are in gut-associated lymphoid tissue. And of course, there is very direct evidence that human milk oligosaccharides do influence the microbiome, specifically and uniquely being the preferred substrate for beneficial microbes. But whether and we do know that the HMOs are absorbed, they can be measured in the blood. So either possibility or both exist.
Okay, great, thank you. Next question is, how can we develop formulas that better support the intestinal microbiome? Could probiotic elements be added to formula if FDA-approved probiotics become available?
Well, certainly they're being studied, not in infant formula. And in the case of preterm infants, that's unlikely because preterm infant formulas have to be liquid, and probiotics would not be able to survive the manufacturing process of sterilization. Conceivably, it would be possible to add them aseptically. So some liquid products are produced aseptically, where the liquid is sterilized and then added aseptically to containers. I mean, you've seen this on the shelf, the cardboard containers that contain soy milk and cow milk, those are all aseptically filled. And there are studies now with probiotics in preterm infants, but not as part of a preterm formula, but rather added as a supplement. But certainly that's an area that's getting a lot of attention. It's almost impossible to pick up a journal of any science journal without seeing one or more papers on some aspect of the microbiome. And the challenge is that there are, within the probiotic arena, there are so many different combinations of species available that it's difficult to know exactly which one may or may not have a specific effect. So when evaluating these probiotics, one has to look at the individual species and make the decision on which ones are creating the desired effect.
Great, thank you. Next question is, please review the amounts of whey and casein protein in mother's own milk over time from birth, donor milk, and preterm formulas, and are the essential amino acid concentrations in human and bovine whey protein the same?
Okay.
That's a loaded question.
Well, let me answer that as simply as I can. It's generally thought that human milk is roughly a 60 to 40 whey to casein ratio. But actually the whey to casein ratio in human milk is quite variable. And it varies between mothers and over time. It's possible to see some mothers' milk that are actually casein predominant. Cow milk has different whey and casein proteins. The major whey protein in cow milk is beta-lactoglobulin that doesn't exist in human milk. All milk proteins are rich in essential amino acids. There are differences in the ratios between whey and casein. Whey, for instance, is very high in threonine. So you do see those kinds of differences. But it's very difficult to make generalized statements about the whey and casein composition of either human milk or cow milk because there's so much variability. I hope that answers your question.
And then we have one more. I know we're a little over time, but we have one more question. Are there animal models that serve as appropriate preclinical models for the preterm infant?
There are a number of animal models. Neonatal pigs are probably about as close to the human infant as we see. But with any animal model, you can never completely assume that the response in humans is going to be exactly the same. So it can give you guidance and give you direction, but ultimately the test has to be in human infants.
Great. Well, I want to thank you very much for your time and your insights. If anyone is interested in receiving references or a PDF of Dr. Maser's presentation, please reach out to leah@astardymedical.com and we'd be happy to share that with you. Thanks, everybody, and please join us for our next webinar on November 17th. Thank you so much.
Thank you.
Yep. Bye-bye.